we'll start in a few minutes. Right, so I'll start by sharing my slides. Right. Welcome to the 12th live session of structural biology. Uh, in this, we will be talking all about drug design. So this is the introduction to drug discovery. Drug discovery is the process by which new candidate medications are discovered. The principle of drug discovery is to create large changes in biology through small changes in chemistry. The principles of drug discovery include selecting a target, like maybe there is a certain defective DNA that we are targeting. Then lead discovery, meaning the initial molecule um, which will be further refined and improved to form the final drug. Then using medicinal chemistry to refine and improve the lead till it has the best results on the target. Then several in vitro and in vivo studies are done till the final drug is obtained. Now we'll briefly look into the chemistry of DNA. So as you know, DNA is a polymer of nucleotides, and each nucleotide consists of a 5-membered sugar. Since it is 5-membered, it is called a pentose sugar. Also, there is a nitrogenous base and a phosphate group. Now, this nitrogenous base can be of 4 types: adenine, thymine, guanine, or cytosine. Now, in DNA, the certain nitrogenous base pairs with other nitrogenous base, and this complementary base pairing is fixed. So adenine and thymine pairs with each other through 2 hydrogen bonds, and guanine and cytosine pairs with each other through 3 hydrogen bonds. These form the backbone of the DNA, and it is the hydrogen bonding between base pairs that gives DNA its stability. Now we will be talking about nucleoside analogues, or Na. This is an example of small change in chemistry which greatly affects biology. So nucleoside analogues are compounds with identical hydrogen bonding properties of natural nucleosides. However, when they are incorporated into a DNA chain, they stop the synthesis or extension of the DNA chain and lead to cell death. So basically, if you look at these structures, at this R position, if there is an OH, it is an adenosine. But if there is an H, it is a 2' deoxyadenosine or a nucleoside analog. So nucleoside analogs often do not have this 2' OH, and that is why if they are incorporated into the DNA chain, the extension of the DNA chain will stop, because DNA chain extension occurs through this 2' OH group. Similarly, in guanosine, if the R is OH, it is guanosine, and if it is H, it is the nucleoside analog deoxyguanosine. Similarly, for cytidine and thymidine. Here, the nucleoside analogs are working as a drug because a small change in chemistry, that is converting this R from OH to H, which is a small change in chemistry, is greatly affecting biology because it is stopping the DNA replication and leading to cell death. So there can be several different types of nucleoside analogs. One is simply removing the OH group, as we discussed. Another type of nucleoside analogs can be taking the L isomer instead of the D isomer. So if you look at the original structure, the O is pointed upwards and the OH and R are pointed downwards. Instead of this, if the orientation is reversed, then it forms a L nucleoside analog. The third kind of nucleoside analog is if instead of the cyclic 5-membered sugar molecule, there is an open chain sugar molecule. So all these will eventually lead to termination of the DNA replication and hence cell death. Now we will briefly talk about finding the lead compound. So a lead compound is a small molecule that serves as the starting point for an optimization involving many small molecules that are closely related in structure to the lead compound. So what this means is that suppose we want to, one second, yes, suppose we want to find a lead compound. So we will examine several biological active compounds found in plants or herbs, and also different marketed products, that is drugs. Through a combination of this, a library of compounds is created and the data is collected to form a library of possible new drugs. Then several in vitro and in vivo studies are carried out, and if any synthetic or chemical modification is required, then that is done to find a group of biologically active compounds. These can now these form the best of this biologically active compound is the lead compound, and the lead compound can now be modified in several ways, and its toxicologic and pharmacokinetic parameters studied through application of drug delivery methodologies. So what this means is that it is not enough for the lead compound to be biologically active. It also needs to be non-toxic so that it is safe for human consumption. Secondly, by pharmacokinetics, it means that it has optimal absorption, digestion, and metabolism in the human body. So after passing through these tests, the optimized therapeutic agent is procured and then sent for clinical trials. Now, many organizations maintain databases of chemical compounds. Some of these are publicly accessible, while others are proprietary databases, meaning that they are private and they contain extremely large number of compounds. For example, the ACS database contains 10 million compounds. Now, if you look at how a lead is found, so from the initial library of compounds which have binding affinities, say around 10 micromolar, all these processes are done, like chemical treatments and pharmacological or toxicological studies. And you can see the final lead compound has a binding of less than 10 nanomolar. So the binding is gradually improved from the initial library of 10 micromolar to less than 1 micromolar, then less than 100 nanomolar, and finally less than 10 nanomolar. And the several lead candidates are then tested for activity. So if you can see in this activity chart, the blue rectangles indicate low activity, while the maroon or brown rectangles indicate high activity. So other than binding, the activity is also tested to find the lead compound. Now we will talk about traditional drug discovery. Traditional drug discovery involves the origin of drug discovery that evolved in natural sources, accidental events, etc. It was not target-based and not systemized or logical like current is. The first type of traditional drug discovery is called random screening. Random screening involves a collection of synthetic compounds or chemicals or natural products which could not be assayed by experimental assay method. It involves two major approaches. Firstly, screening of a selected class of compounds like alkaloids, flavonoids, or any synthetic library collection, etc. The second method is screening of randomly selected, traditionally identified medicinal plants like Ayurveda. So if you can see what random screening does is that it will start with around 5,000 to 10,000 compounds, which may be common medicinal plants or selected classes of compounds which are known to have medicinal properties. After the initial screening is done, which takes 3 to 5 years, it is reduced to 250 compounds for preclinical trials. This takes 1 to 2 years, and then 5 compounds remain for clinical trials, which takes 6 to 7 years. Finally, one new drug is obtained, which is the best in terms of both activity and efficiency, and also non-toxicity and pharmacokinetics. And the approval of this new drug by the FDA takes 1 to 2 years. The second method is called a trial and error method. Trial and error includes berries, roots, leaves, and barks that could be used for medicinal purposes. For example, cinchona bark contains quinine used to reduce fever in malaria. Similarly, leaves of the neem plant are used as anti-inflammatory and antibacterial properties. So in trial and error method, what is done is that a medicinal plant which has potential of being used for medicinal properties. So the medicinal component or chemical compound of that which is responsible for its medicinal properties is extracted, isolated, and purified, and then it is characterized, and finally clinical trials are done to convert it into a ingestible drug and approve it by FDA. So trial and error method naturally takes a lot of time because it is many plants have to be screened to get a viable chemical compound which has potential to be used as drug. The third kind of traditional drug discovery is known as ethnopharmacology approach. Ethnopharmacology is the study of medicinal plants used in specific cultural groups. It involves the observation, description, and experimental investigation of indigenous drugs. For example, Andrographis paniculata was used for dysentery in ethnomedicine, and the compounds responsible for the activity were found to be andrographolites, which are basically used to treat dysentery. So in ethnopharmacology, what happens is that the history and culture of a particular civilization gives information about the kinds of medicinal plants used by that civilization. And from that medicinal plants, the particular compounds responsible for the medicinal activity can be isolated and then tested. The fourth kind of traditional drug discovery is called classical pharmacology or function-based approach. The principle of this is that there is no prior identification of a drug target. Anciently, drug discovery processes were often based on measuring a complex response in vivo, such as prevention of experimentally induced seizure or lowering of blood sugar. For example, discovery of foxglove plant in Europe, which used for drug preparations that contain cardiac glycosides, particularly one called digoxin, extracted from various plants of this genus. So basically, in classical pharmacology, you start with the functional activity in vitro or in vivo. That is, you start with the knowledge that a particular plant has a function in treating a particular disease. Then based on that, the lead compound is isolated, which is the chemical component in the plant which helps to treat the particular disease. And finally, the target is identified. That what is the protein in the body that the lead compound is interacting with to treat the disease. So function-based approach starts with the function, and the final step is identifying the target. The final kind of traditional drug discovery is by serendipitous discovery. So as you know, bacteria was first identified in 1670s by Van Leeuwenhoek, and the relationship between bacteria and diseases was gradually set up in the 19th century. Since that time, researchers have tried to find effective antibacterial agents. It was during this time that penicillin was discovered, and how it was done is that it was completely by accident, and that is why it is called serendipitous discovery. So basically, a bacterial colony was being grown, and it had got contaminated with some penicillin. It was observed that wherever the penicillium was growing, the bacteria growth is inhibited. And wherever there was no penicillin, the bacteria was growing normally. So this was how the world's first antibiotic was discovered, by sheer luck. Now, the time in which penicillin was launched was very lucky that penicillin was found, because it was during World War I, where around 18% of the death was caused by wound-related pneumonia. And after penicillin was found, this 18% death was reduced to less than 1%. So in traditional drug discovery, the scheme of traditional drug discovery is firstly finding a lead, either through some natural ligand or by screening, then doing biochemical testing to find out the chemical mechanism by which the lead is interacting with the protein in the body, then biological testing to see if it is biologically viable, that is, it is non-toxic, and its absorption, digestion, and metabolism is all properly happening in the body. If the biological testing is promising, it can go for preclinical trials. But if it is not promising, then new compounds have to be synthesized from the lead compound. And again, the biological testing has to be done till it becomes promising and can be sent for preclinical trials. There are several challenges in this method of drug discovery. Firstly, the process of bringing a potent drug to market needs a huge investment in time and money. But almost 70% of the funds are wasted due to failure to pass through the clinical trials due to a lack of efficiency or adverse side effects. So although that much time and money is being spent, there is a 70% chance of failure. Secondly, the time and cost enhancement pushes the company to increase the price of market drugs. But due to intense competition and limitation of affordability to common consumers, it is difficult to work with that strategy also. So what this means, in order to make up for the large amount of time and money, the companies are forced to increase the cost of the drugs. But there is no point in keeping on increasing the cost of the drugs, because then the common public will not be able to afford it. And if no one buys the drugs, then obviously the companies will also not prosper. So increasing the cost of the marketed drugs is also not a solution. So these are challenges that are faced in traditional drug discovery. The next kind, which is more commonly used now, is called rational drug discovery. Rational drug discovery can broadly be divided into two major groups. The first kind is called ligand-based drug discovery. So in this, the 3D structure of the biological target is unknown, and a set of geometric rules and or physical chemical properties, that is pharmacophore model, obtained by QSAR studies are used to screen the library. Now, QSAR, as you know, is quantitative structure activity relationship. So basically what happens in ligand-based drug discovery is that you start with a library of possible drug-like compounds, and then you filter the compounds according to some rules, which we will learn about later. These rules basically classify that which of these ligands have a good chance of becoming drugs. Then the pharmacophore modeling is done. That is, the geometry and the physical chemical properties of these ligands are given certain numerical values to denote that which of these have high activity. This is followed by QSAR analysis. That is, the pharmacophore modeling gives you the structure. And then from the structure activity relationship is found out in order to predict that which structure will have the highest activity. Then molecular docking is done. That is, it is a software which will predict the binding affinity of the ligand and the protein. After that, screening is done to select those ligands which show the highest predicted binding affinity from the molecular docking. Then different targets are tried to see whether the ligand has is specifically targeting one particular molecule, or it has it can target many molecules like a broad spectrum antibiotic. And finally, biological assay is done to find how viable it is in the biological system. The second kind is called structure-based drug discovery. So in this, what happens? It involves molecular interaction calculations between each molecule to be tested and the biological target, which is usually a protein. To evaluate the affinity, a scoring function is applied, which we learn about later. And in this structure-based drug discovery, the 3D structure of the target must be known. So as you can see, it starts with the molecular interaction between here you can see this is a protein and the highlighted portion is the ligand. So using this, there is a software which will predict the binding affinity. It will give a score to the binding. The higher the score, the better the affinity. And then this idea can be used to give rise to potential drugs. So that is structure-based drug discovery, where the 3D structure of the target is known. So now we will see a case study of rational drug discovery through which is cimetidine or Tagamet. So the problem was that histamine triggers release of stomach acid. This is the structure of histamine. So a histamine antagonist is required to prevent the stomach acid release by histamine. So that is the biological target. Several histamine analogues were synthesized with systematically varied structures by chemical modification, and the histamine analogues were screened. It was found that N-guanine histamine, which is this compound, where a guanidinium group has been attached to the amino terminal of histamine, and this compound showed some antagonist properties. That means it reacts with histamine to neutralize the release of stomach acid, which is what we want. So this N-guanine histamine was chosen as the lead compound. Then chemical modifications were done to the lead compound, which is called lead optimization. So you can see in burimamide, instead of the double bond N, a double bond S was introduced. And in thiaburimamide, sulfur was introduced previously as well. Then after this lead optimization was done, the next step was which is most potent and orally active, and that was meteamide. But the problem was this thiourea group that is there, NH single bond C double bond S single bond NH. This thiourea group is toxic. It was found to be toxic in clinical trials. So the next step was to replace this group by an effective and well-tolerated product, which gave rise to cimetidine, where this azide group was introduced. And eventually cimetidine was improved even further to form this compound, which is ranitidine or Zantac, which has an even more improved safety profile. So this was an example of rational drug discovery. Now we will study about ligand-based drug discovery. So one of the, there are several methods in ligand-based drug discovery. The first one that we will study is quantitative structure-activity relationship or QSAR modeling. So quantitative structure-activity relationship models or QSAR models are regression or classification models used in chemical and biological sciences and engineering. QSARs are mathematical relationships linking chemical structures with biological activity using physicochemical or any other derived property as interface. So how does this work? The basic assumption is that there is a mathematical function of the chemical properties which is related to the effect. That is y equal to fx. That means there is some structure, you can consider the structure as x and its effect as fx. So suppose the ligand that you are studying, it has some structural feature x, which gives rise to the activity, the drug-like activity that we want, which is denoted by fx. But how to find this mathematical algorithm fx? Typically, we use a number of chemical compounds with known values of the toxic effect y. For each chemical compound, we calculate a series of parameters called chemical descriptors. So basically, for each ligand, there is a series of X, which are the molecular descriptors. That is the physicochemical properties of the molecule. And for each X, we will get a Y, that is the response variable. Meaning for a certain physicochemical property, what is the activity or what is the predicted toxicity. So Y is what determines the potential of the ligand to act as a drug. Then we find an algorithm that provides a quite accurate value similar to the real experimental value. The final step is to check if the so obtained algorithm is capable to predict the property values for other chemicals not used to build up the model. So what this means is that firstly, a structured activity relationship is found. So we have a set of X, that is the molecular structure descriptors, and a set of Y, that is the activity corresponding to each structure. So we can correlate these to find a structured activity relationship. And in order to validate whether our structured activity relationship is correct, a chemical is used which is not the test chemical. So the test chemical is the one which we had used to make the initial structured activity relationship. Now, if we use a chemical unrelated to this test chemical and we predict the values, activity values of that chemical based on its structure from the structure activity relationship, and then the experimentally obtained and the predicted activity values of the new chemical are compared. So if the predicted values are close to that of the experimentally obtained values, then that is called validation of the QSAR model. That is, our predicted structure activity relationship is correct. It is very important to generate a model which is working not only with the chemical substances used within the training set, but also for other chemicals. There are several aspects of QSAR which we'll go through one by one. Firstly, our molecular descriptors. These are simple rules for describing some aspect of a molecule, maybe its structure or its property. Now, 0D descriptors are generally the molecular mass, number of atoms, or atomic mass. 1D descriptors encodes for substructural fragments, like it will encode for, say, the benzene ring will have a certain numerical value. A carboxylic group will have a certain numerical value. So it identifies the fragments as a whole. 2D descriptors use the atoms and connection information of the molecule. So it will denote that which are the atoms and how they are connected, like by single bond or double bond. Then internal 3D descriptors use 3D coordinate information about each molecule. However, they are invariant to rotation and translation of the conformation. So 3D descriptors can be of two types. Internal 3D descriptors, which will have the XYZ coordinates of each molecule, but it will consider the molecule as a whole. That is, the internal 3D descriptors are not affected by rotation or translation of the molecule. Then we have external 3D descriptors, and they also use 3D coordinate information, but they require an absolute frame of reference, like if the molecules are docked into the same receptor. So basically what this means is that external 3D descriptors are XYZ coordinates, and they will change with the rotation or translation of the molecule. Then there are 4D descriptors, which basically classify the intermolecular interactions. These can be of different types, like constitutional or, like there is another way of classifying descriptors. Firstly, constitutional or information descriptors, which include the molecular weight, number of different chemical elements, number of hydrogen bonds or double bonds. Then is the physicochemical descriptors, like lipophilicity, meaning how hydrophobic the molecule is, or polarizability. Then we have topological descriptors, which describe how the atoms are branching. Then there are electronic, geometrical, and quantum chemical descriptors, that is describing the electronic properties of the molecule. And finally, there are fragmental or structural key. That is, if there is some aspect of the structure, which is basically like the 1D descriptors, that is, it defines like substructural fragments, like the carboxylic group or an ester group or the benzene ring. So that is another way of describing the molecule, which is by its different active fragments. Now, in QSAR, there are certain assumptions that are taken. Firstly, the effect is produced by the model compound and not its metabolites. So QSAR focuses solely on the model ligand and not its metabolites. The proposed conformation is the bioactive one, which is not always the case, because often what happens is after metabolizing the metabolite that is obtained, that is the bioactive compound. But one of the assumptions of QSAR is that we assume that the proposed conformation itself is the bioactive one. The binding site is same for all modeled compounds. So it is only if we consider the binding site to be the same that we can get a proper structure activity relationship. That is why it is one of the assumptions that the binding site is same for all the modeled compounds. The bioactivity explains the direct interaction of the molecule and the target, which may not always be the case. There may be several steps between the interaction of the molecule and the target, but it is considered a single step in QSAR. Finally, the pharmacokinetic effects, solvent effects, and diffusion or transport are not under consideration. So in QSAR, what happens is that you have the input ligand or the lead. Now, it can change its structure by different chemical activities, like it can undergo tautomerism or it can undergo protonation. So in protonation, what is happening? An H plus is adding on to this nitrogen. Or in tautomerism, what is happening? This NH3 plus so the double bond is shifting from the N double bond to the NH2 plus double. Now, so it can have different conformations. Now, for each conformation, there is a descriptor. So we learned about the molecular descriptors in the previous slide. So each conformation is described by a set of descriptors, which define its surface potential, its electronics, etc. And the descriptor value is given a numerical value. Then the activity is checked to find the binding affinity and kinetics in order to screen the ligands. And so using the training set of ligands, a structure activity relationship is found. And once the structure activity, you can see the structure activity relationship. So using a training set of ligands, a structured activity relationship is found. And then using an unknown ligand, structure of the unknown ligand, the activity of the unknown ligand is predicted. And it is checked with the experimentally obtained activity of the unknown ligand. And if those two are close to each other, then our QSAR model is correct. Now, topological descriptors, we learned about this in the previous slide. So topological descriptors define the atomic branching. Now, there's a certain aspect of topological descriptors that we need to go into more depth about. So basically, in chemical graph theory, the Wiener index is defined as the sum of the lengths of the shortest paths between all pairs of vertices in the chemical graph, representing the non-hydrogen atoms in the molecule. So what this means is that if you have a molecule, you represent each of the non-hydrogen atoms by a point. And then you take the length of the shortest path between all the pairs of these points. The sum of these lengths form the Wiener index. Now, if you have a plot of the Wiener index versus the boiling point of alkenes, You can see that as the Wiener index increases, which means basically as the molecular weight of the alkanes increases, the boiling point also increases, which is expected because with higher molecular weight, the surface area of the alkanes increase, and this leads to higher van der Waals forces. Now, we will talk about 3D QSAR, which is basically a receptor surface model. So, hypothetical receptor surface model constructed from training set molecules 3D shape and activity data. So 3D QSAR is basically it also predicts the activity, but it considers the structure in terms of the molecule's 3D shape. The best model can be derived by optimizing various parameters like atomic partial charges and surface fit. Descriptors like van der Waals energy, electrostatic energy, and total non-bonded energy can be used to derive series of QSAR equation using statistical method. Now we will shortly talk about the different advantages and disadvantages of predicting biological activity with QSAR modeling. Now, firstly, the advantages. Firstly, QSAR is able to predict activities of a large number of compounds with little to no prior experimental data on activity. So how it does that? From QSAR, you get a structure activity relationship. Now, if you extrapolate that relationship, you should be able to predict the activity of an unknown structure. So to get the initial QSAR relationship, often experiments need to be done on few molecules of a training set, or sometimes even the experiment does not need to be done. It is enough to, there's a library of information on different ligands, and those can be plotted to find the initial QSAR relationship. Secondly, QSAR can reveal which molecular properties may be worth investigating further. So what this means is that QSAR gives an idea that which are the molecular properties that are most affecting the activity. So it narrows down the molecular properties that need to be tuned in order to optimize the activity. QSAR is regarded as green chemistry approach since chemical waste is not generated when performing in silico predictions. So QSAR is mostly dependent on computational predictions, and so it is a green chemistry approach. In vivo and in vitro experimentation can be very expensive and time consuming. QSAR modeling reduces the need for testing on animals and or cell cultures, and so both time and money is saved. Now, there are certain disadvantages of predicting biological activity with QSAR. Firstly, it does not provide an in-depth insight on the mechanism of biological action. QSAR is totally based on a correlation between structure and activity. So there is no in-depth insight on the mechanism of biological action. So what happens is, due to this, there is risk of sometimes of highly inaccurate predictions of pharmacological or biological activity. Since it is a simple linear correlation, often the predictions may be highly inaccurate. Just give me 2 minutes, please. Now we will talk about ligand-based drug discovery. There are two kinds of ligand-based drug discovery. Firstly is the pharmacophore development. So what is a pharmacophore? A pharmacophore is an abstract description of molecular features that are necessary for molecular recognition of a ligand by a biological macromolecule. A pharmacophore model explains how structurally diverse ligands can bind to a common receptor site. Furthermore, pharmacophore models can be used to identify through de novo design or virtual screening novel ligands that will bind to the same receptor. So let us talk about how this pharmacophore development procedure works. So firstly, the binding site in the protein is identified. So suppose the binding site as shown here has 2 hydroxyl groups and 1 carbonyl group. Next, hydrogen bonds are detected which can bind to the binding site of the protein. So if there is a hydroxyl OH, a carbonyl O will bind to it. Similarly, for this hydroxyl OH, a carbonyl O will bind to it. And for the carbonyl O, a hydroxyl OH will bind. So basically, this is these 3 groups are needed in the pharmacophore where there are 2 carbonyls and 1 hydroxyl group in this particular orientation. So this is a pharmacophore because it is describing the molecular feature that is needed for binding to the ligand, binding to the biological macromolecule. Now, chemical compounds which contain this pharmacophore are possible leads. Like 2 examples are shown here of chemical compounds which have 2 carbonyl groups and 1 hydroxyl group oriented in this kind of trigonal orientation. So these molecules are the possible leads. Typical pharmacophore features include hydrophobic centroids, aromatic rings, hydrogen bond acceptors or donors, cations, and anions. A well-defined pharmacophore model includes both hydrophobic volumes and hydrogen bond vector. So like you can see, the pharmacophore features here are 2 hydrogen bond acceptors and 1 hydrogen bond donor. Similarly, there can be other kinds of pharmacophores, like this is a 3-point pharmacophore because suppose it requires a hydrogen bond donor, a hydrogen bond acceptor, and a hydrophobic moiety. So it contains 3 features, and so it's a 3-point pharmacophore. This is a 4-point pharmacophore because it has a hydrogen bond donor, a hydrogen bond acceptor, an aromatic ring, and a hydrophobic moiety. So it has 4 total features for binding to the biological macromolecule, and so it is called a 4-point pharmacophore. These features need to match different chemical groups with similar properties in order to identify novel ligands. Ligand receptor interactions are typically polar positive, polar negative, or hydrophobic. So what is the pharmacophore concept? The pharmacophore concept is that specification of the spatial arrangement of a small number of atoms or functional groups. With the pharmacophore model in hand, databases are searched for molecules that fit this spatial environment of functional groups. So in pharmacophore development, the model needs to be developed, and there are several steps that go into this model development. Firstly, select a training set of ligands. It means choose a structurally diverse set of molecules that will be used for developing the pharmacophore model. Like you saw in the previous slide, that there are certain features that the lead molecules must have, which is called the pharmacophore features. And the library is searched for molecules which have these features to find the possible leads. So it is finding these leads that is called finding the set of ligands which have the pharmacophore features in order to bind to the biological macromolecule. The second step is called conformational analysis. In this, we have to generate a set of low energy conformations that is likely to contain the bioactive conformation for each of the selected molecules. The third step is the molecular superimposition. Here we have to superimpose all combinations of the low energy conformations of the molecules. Similar functional groups common to all molecules in the set might be fitted. The set of conformations that results in the best fit is presumed to be the active conformation. So basically what is done here is that from the conformational analysis, we have found the set of low energy conformations within which the bioactive conformation is contained. So one conformation from each active molecule is found by the conformational analysis. All these low energy conformations are fitted together. Like, for example, if you have two ligands, both having an aromatic group, then the aromatic groups are fitted together. The positively charged pharmacophores regions are fitted together. Like that, the set of conformations that has the best fit forms the active conformation. The next step is abstraction. Here, the superimposed molecules are transformed into an abstract representation. For example, superimposed phenyl rings might be referred to more conceptually as an aromatic ring pharmacophore element. So what does this mean? This superimposition will give you an idea of what are the pharmacophore features that the ligand needs to have. Like if there is a superimposition of aromatic rings, that means the ligand needs an aromatic ring component at that position. If there is a superimposition of positively charged elements, that means the ligand needs a positively charged element in that position. The final step is validation. A pharmacophore model is a hypothesis accounting for the observed biological activities of a set of molecules that bind to a common biological target. The model is only valid insofar as it is able to account for differences in biological activity of a range of molecules. So the final step, the validation needs to be done that indeed the pharmacophore model that we are proposing, uh, all ligands that fulfill that model has some degree of binding to the biological macromolecule. And that is validation of the pharmacophore model. Now we will talk about structure-based drug discovery or SBDD. And one kind of SBDD is called de novo drug design. So what is structure-based drug discovery? The use of three-dimensional protein structure information in the development of new biologically active molecules is termed structure-based drug discovery or SBDD. Now, de novo means to start afresh, start from the beginning, that is starting from scratch. De novo drug design is a process in which the 3D structure of biological macromolecule, usually a protein, is used to design newer molecules. It involves structural determination of the lead target complexes and lead modifications using molecular modeling tools. So what does this mean? Suppose we have an initial core fragment, which is this imidazole ring, and this benzene ring with a CH3 needs to be added. So it can be added in several different ways. Like it can be added to the ortho position at the position adjacent to the nitrogen, or it can be added to the position which is second with respect to nitrogen. So there are actually 4 positions, 1, 2, 3, 4, in which this benzene ring can be added. So this is de novo drug design, where different fragments are added together to form the uh, predicted ligand or lead molecule. So compounds are built that are complementary to a target binding site on a protein via random combination of small molecule fragments to make a complete molecule with better binding profile. So like you saw here, these random combination of small molecule fragments are put together to get a final structure which has better binding than each of these fragments separately. Now, de novo drug design can pursue both receptor and pharmacophore based approaches independently. In receptor based design, what happens is that based on the binding site of the receptor, the fragments are put together to design a possible ligand. And in pharmacophore based design, what happens is that based on the pharmacophore features, that is, say, an aromatic ring is needed in this position. So based on the pharmacophore features, the possible ligand is designed. There are several steps in de novo drug design, and we'll go through these one by one. Firstly is the generation of potential primary constraints. So these are molecules which set up a framework for the desired structure with the required ligand receptor interactions. They are of two types: receptor based, meaning interactions of the receptor form the basis for the drug design, or ligand based, that is, ligand to the target functions as the key. So the initial step of de novo drug design is setting up several structural constraints that the ligand must have in order to interact with the biological macromolecule. This designing of the ligand constraints can be done based on the structure of the binding site in the receptor or through pharmacophore modeling. The second step is derivation of the interaction sites. A key step to model is to a key step is to model the binding site as accurately as possible. This starts with an atomic resolution structure of the active site. Further constraints follow using positions of hydrogen bond acceptors and donors. So what does this mean? So suppose we have an NH in the binding site, then we can predict that the ligand must have a hydrogen bond acceptor or like a carbonyl group. So it is only when the interaction site is properly modeled that we can figure out more structural constraints for the ligand. The next part is building up methods, which are of different kinds, like growing, linking, lattice-based sampling, or molecular dynamics-based methods. So suppose from the first two points, we have figured out that we need an aromatic ring with an OH group and an cyclic structure with a carbonyl group in the ligand. Now we can link these together, that is, one after the other. They can be attached by they can be attached by covalent bonds to form the final ligand structure, or they can be grown. That means that instead of you can see here that the orientation is maintained according to what is predicted from the pharmacophore model. So building up methods include various ways by which the different pharmacophore features are linked in order to get the final ligand. Then assay or scoring is done, which is basically predicting the binding affinity that the ligand will have to the biological macromolecule. Then there is strategies are searched to improve the ligand binding even further. And this is done through secondary target constraints. The second kind of structure-based drug discovery is called docking-based virtual screening. So virtual screening is a computational technique used in drug discovery to search libraries of small molecules in order to identify those structures which are most likely to bind to a drug target, typically a protein receptor or enzyme. So as this definition suggests, virtual screening has largely been a numbers game focusing on how the enormous chemical space of over 10 to the power 60 conceivable compounds can be filtered to a manageable number that can be synthesized, purchased, and tested. So what virtual screening does is that it significantly reduces the time, effort, and money put into actual experimental screening. Docking attempts to find the best matching between two molecules. Given two biological molecules, it will determine that whether the two molecules are interacting, and if so, what is the orientation that maximizes the interaction while minimizing the total energy of the complex. This can be done by various docking softwares like Autodock. There are certain basic requirements for molecular docking. Firstly, the 3D structure of the protein must be known. The library of small molecules as possible ligands must be known, and the proper protein ligand complex must be selected. So, in short, we'll go through this slide again. In virtual screening, what happens is that from literature and laboratory experiments, there is a library of small molecules. These molecules can undergo QSAR modeling to like from the main library. Using QSAR modeling, we can choose a smaller library of compounds which are then screened virtually. That is, machine learning is used in order to predict that which structures will have the best binding. And after virtual screening has significantly reduced the number of possible ligands, then in vitro and in vivo experiments can be done in order to lead optimize and find possible drugs for preclinical trials. So virtual screening, what it does, it reduces the experimentation to only that of prioritized compounds. And in docking, what happens is that docking is a part of this virtual screening. So basically how the virtual screening is done is that docking is done through docking softwares to find the predicted binding affinities of the small molecule and the biological macromolecule. So that can be done if the 3D structure of the protein is known, if a small molecule library is available, and the proper protein ligand complex is known. So once that is known, different docking softwares can predict the binding affinity between the protein and the small molecule. Now we will talk about the different molecules available for virtual screening. So firstly, there are real molecules, that is 1 to 2 million in-house archives of large pharma and agrochemical companies. And 3 to 4 millions of these real molecules are available commercially. The second are hypothetical molecules, which are virtual combinatorial libraries having up to 10 to the power 60 molecules. Now these small molecule libraries can be divided into 4 groups. Firstly, general libraries are designed to be of broad interest for high throughput screening against any target. Then there are focused libraries which are aimed at a particular family of related targets. For example, a library designed against the chymotrypsin fold or against serine protease inhibitors. Then there are targeted libraries aimed at a single therapeutic target, such as HIV-1 protease or beta-lactamase. And finally, there are cherry-picked libraries, which is basically an exclusive library of a set of compounds, which we can ask a company to synthesize for us. So basically, there are different kinds of libraries as we discussed. There are very selective libraries like the cherry-picked libraries. There can be clinically approved libraries, that is consisting of molecules which have been clinically approved. There are historic libraries which were basically used for the traditional drug discovery methods. There can be semi-selective libraries which are in the stage of clinical development. That is, they have not yet been completely approved clinically, but they are working towards that. And there are poly and unknown libraries, which basically consist of hypothetical molecules where different fragments can be virtually combined to have a hypothetical library of molecules. Now we will be talking about scoring functions. Scoring functions is an approximate mathematical method used to predict the strength of the binding affinity between two molecules. Suppose you have two molecules, and scoring functions will give you the best binding affinity between these two molecules. So it can happen that the molecules are interacting outside the interaction cavity. Then the binding affinity will not be like the energy of binding is quite unstable. Or it can be within the binding cavity, but it is not properly aligned. So again, it is not the lowest energy structure. The lowest energy structure will be that which is the best docking model. And scoring function will predict the binding affinity of this best docking model. Scoring functions are generally classified into three basic types. Force field analysis, which can calculate the actual molecular forces that exist in the protein ligand complex by optimizing van der Waals, electrostatic and hydrophobic forces to each force field. But the problem is it is an extremely complicated process and it takes a long time. Next is the empirical scoring function. This is the fastest scoring function, and it optimizes empirical terms like van der Waals interaction, hydrogen bonding, electrostatic energy, hydrophobicity, desolvation, and change in solvent accessible surface area. The third kind of scoring function is a knowledge-based scoring function, which uses experimentally determined structure geometrical data from databases like the Protein Data Bank. It converts structural information from protein ligand complexes into distinct dependent pair potentials. So basically the three types of scoring functions that are there, one of which are dependent on the force field, and it optimizes each of the interaction forces like the electrostatic and the van der Waals with respect to the force field. In the empirical scoring function, each of the terms are optimized, like the van der Waals interaction term, the hydrogen bonding term, etc. And in the knowledge-based scoring function, knowledge of structural data from protein data bank is used to convert the information of protein ligand complex into distance-dependent pairwise potentials. Now we will talk about enzyme substrate binding. There are two, excuse me for 1 second. Yeah. There are two models of enzyme substrate binding. Firstly is the lock and key model. In this, what happens is that the active site of the enzyme has a fixed structure, and the substrate also has a fixed structure, which corresponds exactly to the cavity in the active site. They come together to form the enzyme substrate complex. The substrate is converted into the product, which is released, and the enzyme is released as it initially was, that is, it is regenerated. In the induced fit model, what happens is that the active site has a certain flexibility, and the cavity changes its size and shape in order to accommodate the substrate. So the enzyme is adjusting its shape. Then again, the substrate is converted to the product and the enzyme is regenerated. Now, one receptor can have several ligands. That is, it can bind to several ligands. Now, if the protein binds to several ligands and the affinity for binding ligands 2, 3 changes after ligand 1 is bound, then the binding is said to be cooperative. Now, if it happens that after binding of the first ligand, the affinity for the ligands 2, 3 increases, then it is called positive cooperative. And if it happens that after binding of the first ligand, the binding affinity of the second, third, fourth ligands decreases, then it is called negative cooperativity. So now, how is the binding predicted? Computationally, lock and key is the simplest case to predict because there is little or no flexibility to be modeled. Like we talked in the lock and key model, both the enzyme and the substrate structure is fixed and it is rigid. So in lock and key, there are only 6 degrees of freedom, that is 3 rotation and 3 translation. The induced fit is much more difficult to model because there are much greater than 6 degrees of freedom. Because in induced fit, as we studied, the enzyme can change its shape and structure. So algorithms need to model the movement of side chain, backbone, and ligands in induced fit. So it is slightly more difficult. Then there is molecular dynamics and receptor flexibility. Relaxed complex scheme molecular dynamics simulation combines all atom nanosecond long molecular dynamics simulation of the protein target in order to describe its conformational flexibility with rapid docking of small molecules to the protein snapshots saved along the MD run. So what does this mean? In order to model the flexibility of the protein, molecular dynamics simulation is done, and snapshots are saved in the course of this molecular dynamics simulation to capture the different conformations of the protein. Then docking is done using small molecule ligands with each of these conformations of the protein to find that which has the best binding affinity. However, often the conformation change needed to uncover the binding pocket would not be observed at the 1 millisecond timescale accessible to explicit solvent molecular dynamics simulation. So in that case, what is done? The molecular dynamics induced fit protocol makes use of atomistic simulations to increase or reduce the aperture of subpocket in the active site. So if by chance the timescale that is required to uncover the binding site is not observed in the timescale of normal molecular dynamics simulation, then a special kind of molecular dynamics simulation called MDIF or molecular dynamics induced fit. So what it does simulations at the atomic level in order to quickly uncover the binding pocket to predict the binding to the active site. So what is a good drug candidate? A good drug candidate will have good activity or selectivity on the correct target, and it will also elicit the desired biological response. And the third point is that a good drug candidate will have proper absorption, distribution, metabolism, and excretion, and it will have no toxicity. So when all these criteria are fulfilled, it forms a good drug candidate. The problem is that to find a good drug candidate, there are 3 stages: research, development, and finally the market stage. And a huge amount of money goes into the research and development stage. Like in the research stage, $300 million and 4 to 5 years is required, while in the development stage, $500 million and 8 to 10 years is required. The failure rate can still be over 80 to 90% due to toxicity or lack in safety or efficiency of the drug. So how to solve this is that identify the problems in absorption, distribution, metabolism, excretion, toxicity in the earlier stages itself, so that it doesn't go to the final stage and you find out that there's a problem in these points. And place emphasis on these points during the lead optimization process itself. That is a way to reduce the failure rate of the final drug. What is the reason for the drug failure in the clinical development? That is where 80% of the drugs fail. There are several reasons. Like you can see, 39% is due to pharmacokinetics. That means the absorption, distribution, metabolism, excretion is not proper. Second, then it can be due to toxicity. It can be because it is causing some adverse side effect. Or it can be due to commercial reason, like the drug is very costly. Or due to lack of efficiency. That is, it is not treating the disease as well as expected. So what are the in silico points that must be considered during drug design? These are the pharmacokinetic parameters, and they are firstly the oral bioavailability. That is the fraction of the dose that enters the blood circulation after first metabolism in the liver. This must be sufficiently high. Only then enough of the drug will enter the blood circulation and finally the affected region. Then the absorption, the fraction of dose that passes the gut wall. This must also be high because otherwise the drug will be excreted without being allowed to carry out its function. Then clearance or CL, which is the amount of blood cleared per unit time. And finally, the volume of distribution VD, which is the IV dose divided by the initial plasma concentration. The volume of distribution must also be quite high so that the drug is distributed throughout the body. Now, the volume of distribution and the clearance will together determine the dosing regimen. That is how often the drug needs to be taken. And the oral bioavailability will determine how much of the drug needs to be taken. Membrane permeation is also important. So it has to permeate through the cell membrane in order to go inside the cell and carry out its activity. So the penetration rate is defined by this formula, which is P into A into C1 minus C2, where P is the partition into the membrane, A is the effective surface area of the membrane, and C1 minus C2 is the concentration gradient inside and outside the membrane. We'll quickly talk about partition coefficient. A partition coefficient or distribution coefficient is the ratio of concentration of a compound in a mixture of two immiscible solvents at equilibrium. In pharmaceutical sciences, both phases are usually solvents. Most commonly, one of the solvents is water, while the second is hydrophobic, like 1-octanol. Like you have benzoic acid in water. It is in equilibrium with the deprotonated state of benzoic acid in water, and this is in equilibrium with octanol. Now, in octanol, the deprotonated state does not exist that much. One second. So this state does not exist in octanol because it is a charged state. And as you know, octanol is a hydrophobic solvent, so it will not stabilize the charged state. So the partition coefficient is defined by log P is log of the concentration of the molecule in octanol by the concentration in water. And log D can be written as log of, in the numerator, concentration of the compound in octanol, protonated and the unprotonated, and again concentration of protonated and unprotonated in water. The partition coefficient measures how hydrophilic, that is water loving, or hydrophobic, that is water fearing, a chemical substance is. So if the chemical substance is hydrophilic, then its concentration in water will be much higher, and it will be the log will become negative. And if it is hydrophobic, then the concentration in octanol will be much higher, and this whole thing will become positive. Partition coefficients are useful in estimating the distribution of drugs within the body. Hydrophobic drugs with high octanol water partition coefficient are mainly distributed to hydrophobic areas, such as lipid bilayers of cells. Conversely, hydrophilic drugs with low octanol water partition coefficients are found primarily in aqueous regions such as blood serum. Now, if you look at the definition of the partition coefficient, you can see that it depends on the pH. So how does the pH change in the human body? The intraluminal pH is rapidly changed from highly acidic in the stomach, because the stomach has concentrated HCl, to about pH 6 in the duodenum. The pH gradually increases in the small intestine from pH 6 to about pH 7.4 in the terminal ileum. The pH drops to 5.7 in the cecum, but again gradually increases, reaching pH 6.7 in the rectum. So depending on the pH, the partition coefficient also varies, and that will determine how much of the hydrophobic or hydrophilic compound diffuses in each of these parts with the different pH. Now, there is a term called clog P, which can be calculated for a particular ligand or structure. So what is done is that this ligand molecule, the isolating carbon or IC are determined, which are the carbons that are not doubly or triply bonded to a heteroatom. So in this particular case, there are 6 such IC. So as you can see, this carbon is doubly bonded to a heteroatom, so it's not counted. So there are 6 IC, and they will have a certain contribution numerical value. The remaining fragments are characterized by topology and environment. So for carboxy carbon, there is only one. Hydroxyl carbon, there are 2 carbons which are directly bound to a hydroxyl. So like that, there are different contributions by different fragments, and clog P is a sum of all the contributions by the fragments plus the isolating carbons plus corrections made due to intramolecular, polar, dipolar, and hydrogen bond or electrostatic interactions. The clog P predicted for this structure is 1.56, and experimentally it is 1.58. So it's pretty close to each other. There is a rule for intestinal absorption called Lipinski's rules or rules of 5. There are 4 points in this, which is basically the number of hydrogen bond donors, that is the sum of OH and NH groups, should be less than 5. The number of hydrogen bond acceptors, that is the sum of N and O atoms without hydrogen attached, like carbonyl or imine, should be less than 10. The molecular weight should be less than 500 Daltons, and log of the partition coefficient should be less than 5. If these 4 rules are followed, then the intestinal absorption is good. So an example is given of this molecule called paclitaxel or Taxol. It was initially used as medicine, but it was later found not to be very good because it violates 2 of the Lipinski's rules. Firstly, the molecular weight is 837, and it should have been less than 500. And the number of hydrogen bond acceptors is 15, and it should be less than 10. So these 2 Lipinski's rules are violated, and so it was later discontinued as a drug because the intestinal absorption was not very good. And it also had a lot of side effects like hair loss, bone marrow suppression, numbness, allergic reactions, muscle pain, and diarrhea. And all these side effects were due to improper intestinal absorption. Now there is a problem that needs to be addressed, and that is called antimicrobial resistance or AMR. What happens is that in the as more and more antibiotics are developed to kill bacteria, you have to remember that bacteria was also created by nature. And so through the process of evolution, bacteria develops a resistance to these antibiotics, and the antibiotics no longer work against the bacteria. So we need even newer antibiotics. So there is a race that is happening between human and bacteria, where the more the humans are developing newer antibiotics, the more the bacteria are developing resistance to these antibiotics. So this is a problem that needs to be addressed. So as you can see that up to 2018, the number of 0.1 million like bacteria have developed resistance against 0.1 million antibiotics. And this is predicted to go up to 10 million in 2050. And this is a very dangerous thing, because without antibiotics, death is certain. But the antibiotics that are there are starting to become less and less effective. Now we will look at a case study that is, just give me 1 minute. We will now look at a case study that is combating antimicrobial resistance targeting enzyme beta lactamase, applying theranostic strategies. So for therapeutics, there are several points. Firstly, designing of new drugs. Then targeting the intrinsic property of some enzyme using these drugs. Then characterizing the enzyme and identifying the newly emergent mutants. So the hypothesis was that every beta lactamase is different with individualistic uniqueness. So the solution is that we have to study each and every beta lactamase coming out of any organism. And this is impossible because there are thousands of beta lactamases corresponding to thousands of genes, and their characterization is impossible. So an interdisciplinary approach has been adopted, combining microbiological screening with computational and experimental techniques. So basically what is done is that identification and genetic characterization of new gene mutants is done experimentally, and a computational database is developed. So both experimentally and in silico, new gene mutants are identified. Then they are cloning, expressed and purified, and the enzyme is characterized. And from that, the new drug is designed, developed, and tested. Now we have to look at an anti-tuberculosis drug development by inhibiting the beta-lactamase with novel boronate transition state analogs, or BATSA. In 2011, 8.7 million people fell ill with tuberculosis. New infections are occurring at a rate of about 1 per second, but standard tuberculosis chemotherapeutics take around 6 months. Now, BLAC or BLAC, the beta lactamase of tuberculosis, has been identified as a major resistance factor. So we ask that, is BLAC a possible pharmacological target to overcome beta-lactam resistance in TB? Indeed it is, because how the beta-lactamase works is that, suppose in active penicillin, this is called a beta-lactam ring. What happens is that beta-lactamase breaks this ring and inactivates the penicillin. So we need a small molecule that inhibits the beta-lactamase. A novel inhibitor, BATSA, was proposed, which has this boronate group. So how this works is that in beta-lactamase, what happens is the serine of beta-lactamases, a serine is present in the beta-lactamase active site, and it attacks this carbonyl of the beta-lactamase and attaches to it. and then it breaks the amide bond, causing hydrolysis and opening of the beta-lactamase. If you have this boronate analog, what happens is that serine, instead of attacking the beta-lactamase carbonyl, will get complexed with the boronate. And so the serine will be blocked from opening the beta-lactamase. So the designing logic of BATSA was that the central boronate interacts with the catalytic serine OH. So what is done is that uh, in the beta-lactamase, the cloning, expression, and purification is done. And then the kinetics is studied using this boronate BATSA ligand. And several BATSA compounds are screened to find a binding of less than 10 micromolar, because that will be that is what is minimum required to inhibit the beta-lactamase. What was found is that 5 compounds were observed with less than 10 micromolar binding, and the best binding, that is the lowest value, which is 0.65 micromolar, which shows the strongest binding, was the one proposed for the anti-tuberculosis drug. This one, called EC19, successfully inhibits the tuberculosis cells. So here, if you have the rate of cell growth and here the number of days, you can see that EC19 is significantly slowing down the growth of tuberculosis bacteria as the days proceed. What was observed is that this benzoic acid moiety that was there is necessary for the effective inhibition, because it is binding, the carboxylate of the benzoic acid is binding with an arginine and a threonine. So these hydrogen bonds help in binding of the ligand to the beta-lactamase, and so this moiety is very important. Understanding the interaction of phenyl carboxyl moiety might help in future for newer generation of drug design. We will look into brief of another project related to the beta-lactamase inhibitor development. This is called the development of DBO moiety-based novel drugs. So diazabicyclooctane, or DBO, are a class of serine beta-lactamase or SBL inhibitors that use a strained urea moiety, which is this N C double bond O N. This strained urea moiety is used as the warhead to react with the active serine residue in the active site of serine beta-lactamases. So like we saw, serine is responsible for breaking the amide bond in beta-lactams. So if we have this strained urea moiety, this serine will attack the carbonyl of this moiety, and it will get trapped by this diazabicyclooctane, and so it will not be able to break the beta-lactam. So this is how the DBO works. So as you can see here, how the DBO NXL104 is a kind of DBO which is inhibiting the beta-lactamase from Mycobacterium tuberculosis. So if you see this at t equal to 0 minutes, the beta-lactamase is completely present. And as you eventually go to t equal to 600 minutes, the activity of the beta-lactamase reduces till it becomes nil, and a new peak appears for the DBO serine complex. So DBO can also be used as drugs for the beta-lactamase inhibition. Now we have gone through the summary of week 12. We'll now go through some sample questions. The first question is that which of the following are processes of traditional drug discovery? So the correct answer is D, that is all of the above. So if you recall, the methods in traditional drug design are firstly random screening. So I'll just go back to the traditional drug screening place. Yes. So the methods of traditional drug discovery are random screening, where a collection of synthetic compounds, like either selected classes of compounds like alkaloids or flavonoids, or traditionally identified medicinal plants, are screened and then experimentally assayed. Another is the trial and error method, where berries or roots that could be used for medicinal purposes are uh, uh, screened through a trial and error method, and the active compound is identified and used in clinical trials. Then is the ethnopharmacology approach, where the medicinal plants used by specific cultural groups are studied, or the classical pharmacology approach, which is a function-based approach, where we start with the known function of a particular plant or compound, and from there we identify the lead compound, and from there we identify the target. And the final is the serendipitous discovery, where completely by luck a drug is discovered, like the case of penicillin. So the five kinds of traditional, one second. So the five kinds of traditional drug discovery are random screening, trial and error method, ethnopharmacology approach, serendipity method, and classical pharmacology. The second question is that which of the following is not involved in rational drug discovery? So the answer is C, trial and error method. So like we saw in the previous slide, trial and error is a method of traditional drug discovery. In rational drug discovery, you have two types of approaches: the structure-based drug design, or SBDD, or the ligand-based drug design, or LBDD. In structure-based drug design, the binding site is identified, and then docking is done in order to give a predicted binding affinity. And using docking, virtual screening is done in order to narrow down the choices of the compounds, and that is finally used for lead optimization and further clinical studies. In ligand-based drug discovery, there are two approaches. One is the QSAR, or the quantitative structure-activity relationship, where using a set of test molecules, a correlation is found between structure and activity, and that correlation is used to predict the activity of a new structure of a new ligand. Or pharmacophore modeling, where from the structure of the binding site, certain chemical features are identified which are essential in the ligand, and those ligands form a library of compounds that fulfill these chemical features from the pharmacophores. These can again undergo virtual screening for eventual compound selection and lead optimization. So rational drug discovery, QSAR modeling forms a part of rational drug discovery. Similarly, pharmacophore modeling also forms a part of rational drugs discovery, and so does virtual screening. So clearly, trial and error method is not involved in the rational drug discovery because it is a traditional drug discovery method. So the correct answer is C. The next question is that what are descriptors in QSAR? So the correct answer is A. Chemical characteristic of a molecule in numerical form. So if you recall, 0D or 1D descriptors are basically how many different kinds of chemical elements there are, or what is the molecular weight or atomic mass. That is 0D. 1D basically refers to the different chemical groups that are present, like say aromatic ring or carboxylic acid or ester. 2D refers to the different atoms and their connectivity. 3D refers to the three-dimensional XYZ coordinates, which can either be internal 3D, which are not changed by the rotation or translation of the molecule, or external 3D, which are changed by the rotation and translation of the molecule. 4D refer to intermolecular interactions. So all these 1D, 2D, 3D, 4D, 0D are represented by certain numerical values, and they form the descriptors. So the chemical characteristics of the molecule, which are in numerical form, are called the descriptors in QSAR. And so the correct answer is A. The fourth question is that atomic branching and ramification fall in which type of descriptor? So the correct answer is topological. So basically, topological descriptors describe the atomic branching and ramification. So if you look at this cuboidal structure, then the green dots with the branching indicate face-centered atoms. The black dots indicate atoms that are bonded to four others. So you can see there are four bonds from each black atom. The green atoms without the branching are other atoms bonded to chain in Q, like this one. And the white atoms are atoms bonded outside of the Q. So like this is a topological descriptor, and it is describing the atomic branching and its consequences. So the correct answer is B, topological. Question 5 asks, what are the physical properties of compounds? So the correct answer is D, all of the above. So like you can see, the physical properties of these ketones are listed, and molecular mass or weight falls within the category of physical properties. So does boiling point, and so does solubility in water. So clearly, molecular weight, boiling point, and solubility are all physical properties of compounds. So the correct answer is D, that is all of the above. The next question asks that what are the basic requirements for molecular docking? So the correct answer is D. So basically for molecular docking, what you need is the 3D structure of uh, the 3D structure of the protein or receptor. And this is generally obtained from databases like the protein databases or by modeling. And a library of small molecule ligands is required. Then the molecular docking can be done. That is, computationally, the small molecule and the receptor uh, can be computationally simulated to find that which small molecule ligand will have the best binding to the receptor. That is called docking. So the basic requirement for molecular docking is to know the three-dimensional structure of the protein or receptor and to have a library of small molecule ligands. So the correct answer is D. The next question is that which of the following is docking software? So the correct answer is B, Autodoc. Autodoc is a software which basically, once the three-dimensional structure of the receptor or protein is inputted and the library of small ligands is also provided, Autodoc will screen the ligands according to which has the lowest binding and uh, most stable ligand receptor interaction, that is the lowest energy, and the highest energy, meaning the most unstable ligand receptor interaction. So using Autodoc, virtual screening can be done in order to narrow down the ligands which will show the strongest binding to the receptor or protein. And then these ligands which have been narrowed down can then be experimentally tested. So the correct answer is B, Autodoc. The next question asks, in virtual screening, hypothetical molecules mean what? So the correct answer is B, library of virtual combinatorial molecules. So basically what happens is that there is a library of molecules which are not a real library, but it is a virtual library because it consists of different combinatorial like if there are different fragments which make up a pharmacophore, then these fragments can be computationally combined and they've also form part of the library. And so it is a virtual library. And these hypothetical molecules can also be used in virtual screening in order to find out that which are the molecules which potentially have the highest binding to a particular protein. So in virtual screening, hypothetical molecules mean a library of virtual combinatorial molecules. So the correct answer is B. The next question is that what is the docking scenario between receptors and ligands for the lock and key model in docking? So the correct answer is A. Now, if you recall, what happens in the lock and key model is that the shape and structure of the receptor and ligand are fixed, and the ligand fits exactly into the cavity of the receptor. So clearly, both the receptor and ligand are rigid. And so the correct answer for the lock and key model is A, rigid receptor, rigid ligand. We'll now go to the 10th question, which asks, which of the following is present in Lipinski rule of 5? Sorry. The correct answer is B, because if you remember Lipinski rules of 5, it states that the hydrogen bond donors must be less than 5, which is the sum of OH and NH. So A is incorrect. The molecular weight must be less than 500 Daltons. So C is incorrect. Log P must be less than 5. So D is incorrect. But the hydrogen bond acceptors, that is the sum of N and O, which is not bonded to hydrogen, must be less than 10. So B is correct. So the correct answer is B. The 11th question is that which of the following approach is considered the ligand-based drug discovery? The correct answer is D. So in ligand-based drug discovery, The types are QSAR, which is basically quantitative structure activity relationship. Then it is pharmacophore modeling. And there are other methods like similarity searching, scaffold hopping, machine learning, and virtual screening. In structure-based drug discovery, there is molecular docking, de novo design, molecular dynamics simulation, and virtual screening. So clearly, in ligand-based drug discovery, pharmacophore modeling is there and QSAR modeling is there. So both A and B is there. But molecular docking is a type of structure-based drug design and not ligand-based drug design. So the correct answer is D, that is both A and B. We go to the last question of today, which asks, which step is not included in the de novo drug designing? So the correct answer is C. So if we quickly look at the different steps in de novo drug design, first is the generation of potential primary constraints, which is A. Next is the derivation of interaction site, which is B. Then is building up methods, that is D. Then assay or scoring, search strategies, and secondary target constraints. So clearly, option C, that is primary target constraints, is not a step in de novo drug designing. So the correct answer is C. That brings us to the end of today's presentation. We will have one more final 1 hour class tomorrow, where it will be a summary of all the 12 lectures. So if you have any doubts, feel free to contact me at my email address. And that is the end of today's class. See you tomorrow.